You have previously learned that you can create transgenic animals to overexpress a gene or use CRISPR-based genome editing techniques to knock out a specific gene. These strategies can help you examine gene functions. They are powerful and informative, but you need to start with a known gene. What if you don't have a candidate gene in mind? In this case, you may consider performing a genetic screen. This is a very useful phenotype-based experimental approach that can help you identify important genes involved in the biological questions you want to address. In this video, I will first use Drosophila pattern formation as an example to help you appreciate the applications of genetic screens. Then I will discuss practical issues that can help you design your own genetic screens. At the conclusion of this video, you should be able to explain how researchers use model organisms to address specific biological questions. You should be able to describe the genetic pathway that regulates pattern formation in Drosophila. You will also be able to choose a suitable model organism, design a genetic screen, and use various genetic tools to answer your specific questions. The fundamental concept of using model organisms is that all living forms, despite their clear differences in their morphology, share similar cellular and molecular features. For example, all eukaryotic cells have a cell membrane, a nucleus, and cellular organelles like mitochondria and ER. They use the same genetic code and very similar metabolic pathways to support cellular structure and function. Importantly, the sequences and functions of most homologous proteins are highly conserved among species. So the knowledge we learned from one model organism could be generalized to other species, even humans. Genetic screens have contributed significantly to our understanding of the concept of genes. Beadle and Tatum's study led to the one gene, one enzyme hypothesis. Benzer's study using the R2 locus of bacteriophage had taught us that genes are composed of a linear array of mutable sites. Now we know they are nucleotides. And Francis Crick's suppressor screen helped to break the genetic code. These studies brought genetics to the molecular level. Today, we will look at how genetic screens in Drosophila help us understand the molecular processes that transform a fertilized egg into a complex organism, with all cells assume correct identity and morphology, and all organs form in the right place at the right time. This story started in the late 1970s when Jannie Nussan-Volhard and Eric Weschhaus investigated events in development that determined the spatial organization of Drosophila embryos. They choose Drosophila as their model organism because the fruit flies are small in size, have short life cycle, and can produce large number of progeny. They are mutable and have a small genome size. These are important features for genetic experiments. The Drosophila development involves a series of morphogenic events that transform a fertilized egg into a segmented embryo. Now we call this process pattern formation. The identity of these segments is preserved to the adulthood. The anterior segment will form the future head and thorax. The posterior segment will become the future abdominal segment. The researchers generated mutants with defects in the development of these segments. They later cloned those genes that are responsible for the mutant phenotypes. Their work let us understand that the body plan is established by tightly regulated sets of transcription factors and signaling molecules produced by the mother and the embryo itself. One of the most striking mutant phenotypes is resulted from mutations in a gene called bicoid, which means two tails. In the bicoid mutant embryo, the segment where a head should be is now a mirror image of the abdominal segment. Bicoid is a maternal effect gene, which means the gene activity is determined by the mother, not the embryo itself. All progenies of two heterozygous bicoid flies has a normal pattern. All progenies of homozygous male bicoid flies develop a head. However, all progenies of a homozygous bicoid female fly have two tails, no matter what their genotypes are. Because the bicoid phenotype is determined by the mother's genotype, scientists call this type of genes the maternal effect genes. Now we know that the female flies make bicoid messenger RNA and transport it into the anterior pole of a developing egg. After the eggs are laid and fertilized, the maternally deposited bicoid messenger RNA at the anterior pole is now translated into bicoid protein. The bicoid protein then forms a concentration gradient with highest concentration at the anterior end and consequently pushes this region to become the head segment. The posterior pole, on the other hand, is exposed to a very low concentration of bicoid and a very high concentration of another maternal effect gene called nanos and develop into an abdominal segment. Homozygous recessive bicoid female cannot produce normal bicoid. Therefore, their eggs do not have bicoid messenger RNA and consequently no bicoid protein. And this kind of embryo cannot develop a head. Now we know that the bicoid protein is a transcription factor that regulates the embryo's own developmental genes. Together with other maternal effect genes, bicoid regulates the expression of a cascade of transcription factors and signaling molecules in different parts of the developing embryo, which will then determine the identity of each segment of the body. Importantly, these transcription factors and signaling molecules are not only used to regulate early pattern formation in the fruit fly. They also have critical regulatory roles in different cell types later in development and in adults in many species. For example, hedgehog and windlass are two signaling molecules that are expressed in different domains of each segment. This diagram shows two segments of Drosophila embryo. Each segment can be further divided into two parts, the anterior part and the posterior part. Hedgehog expression is restricted to the posterior half of each segment, whereas windlass is expressed in the anterior half. Together, these genes help to define the anterior and posterior polarity within the embryonic parasegment. Later in development, during the pupal stage, hedgehog expression is reactivated in a region called the ring disc. This is the larval tissue where the future wing will form. This image shows that hedgehog expression is restricted to the posterior half of the wing disc. Interestingly, hedgehog expression is also activated in the embryonic region of a chick embryo called the limb bud, which will form the future limb. Similar to what has seen in Drosophila wing disc, hedgehog expression is restricted to the posterior end of the chick limb bud, and its protein forms a concentration gradient. Tissue exposed to the highest concentration of hedgehog signaling becomes digit 3, and those exposed to the lowest concentration of hedgehog become digit 1. If you ectopically express hedgehog in the anterior end of the limb bud, you will create a mirror image of digits. Similar regulation is also seen in humans. Abnormalities in hedgehog signaling in human alter normal limb development. To conclude, now you have seen how genetic screens in Drosophila helped us understand the molecular pathways that establish the body plan and how these genes are repeatedly used in different cell type, different biological processes at different developmental time in different species, including humans. In the second part of this video, I will discuss practical issues involved in designing a genetic screen.